right. Hi, I'm Marian, and I am representing the affirmative side. I'm the second speaker. So first, I would like to restate the claim that we made that scientific research is harmful to animals and is humane, because each year more than 100 million animals, including mice, rats, frogs, dogs, cats, rabbits, hamsters, guinea pigs, monkeys, fish, and birds are killed in the U.S. laboratories for biology lessons, medical training, curiosity-driven experimentation, chemical, drug, food, and cosmetic testing. And this is reported by the U.S. Department of Agriculture, Animal and Plant Health Inspection Service. So in fact, animal testing is useless because experiments are flawed. Quoted from Daniel Hackman and Donald Redemeyer, the differences in anatomy, metabolism, and physiology for testing animals can cause results in can vary greatly between human and animal testing subjects. A common example is the active ingredient of Tylenol, which is acetaminophen. It is poisonous to cats, but a reliever to pain for humans. Many examples such as this exist, including the differing results in the same species of animal. These differences become more apparent in the variations of sex, weight, range, breed, and as well as age. Scientists think that scientists believe that testing on animals is better because they have similar genetic makeup or biological processes, but in fact that's actually not true. Quoted from the Anti-National Vivisection Society, although this methodology has become more the norm in research labs around the world, a closer look at the data makes it clear that species who share closer evolutionary relationships or anatomical similarities do not necessarily undergo the same biochemical mechanisms or physiological responses. In fact, it can be dangerous to apply to animal-derived data to humans. For example, from the same source, the National Anti-Vivisection Society, rats and people differ right down to the pH level of saliva. Rats cannot vomit, because they don't have a gallbladder. The drug tamoxifen was patented as a contraceptive, which it is in rats, but in women it has the opposite effect. A man-sized rat could consume 12 bottles of Scotch whiskey a day with no more liver damage than it caused a half bottle in the human liver. Another example is that 94% of drugs that pass an animal test fail in human clinical trials. According to neurologist Asia Akhtar, over 100 stroke drugs that were effective when tested on animals has failed in humans, and over 85 HIV vaccines failed in humans after working well in non-human primates. A 2013 study published in the Proceedings of the National Academy of Sciences of the United States of America found that nearly 150 clinical trials of treatments to reduce inflammation in critically ill patients have been undertaken, and all of them failed, despite being successful in animal tests. Another 2013 study in the Archives of Toxicology stated that the low predictability of animal experiments in research areas allow direct comparisons of mouse versus human data, puts strong doubt on the usefulness of data as a key technology to predict human safety. Animal testing is useless because experiments are flawed. Even scientists are aware of the inherent laws that come with using animals as models of human disease. They recognize that no one can replicate the human condition. Although they have discovered some animal models which appear to mimic parts of what occurs in humans and conclude that they have a good model, the truth is a so-called recapitulation of the human condition may arise by different mechanisms in animals due to the intrinsic differences between species. There is very little consensus among researchers about the right animal model to use in many instances, because in reality, there is no such thing. The law doesn't regulate animal testing. The Animal Welfare Act and the policy Policy on Humane Care and the Use of Laboratory Animals are two main regulations governing the use of animals in U.S. labs. However, even with the supposed protections favored by the AWA and the PHS policy, millions of animals suffer and die in unnecessary, inadequately monitored research labs. The act is not, according to the explanation of the Federal Animal the Federal Animal Welfare Act, this act is not intended to regulate how animals are used in research purposes, but only set standards for how they are obtained and maintained at a facility. When the Animal Welfare Act was amended in 1970, the definition of animal was expanded to include warm-bodied general animals for the use of research, testing, experimentation, exhibition, or as pets, but it clearly excluded farm animals, including horses, livestock, and poultry, while mice, rats, birds are now clearly within the coverage of the Animal Welfare Act as warm-blooded animals. The USDA made a calculated decision to omit them when they are drafting regulations to implement the law. So pretty much what that means is that the Animal Welfare Act covers every single other animal but rats, rats, mice, and birds. So millions of animals still die when being used in animal testing, and it is unnecessary because there are alternative ways.